So annoyed. Okay, so let me give you guys a couple of announcements before we start material. Important things that you will want to know. Where are they? Oh, okay. You ready? You listening? Okay. Today at 4:30, if you happen to be on campus or near campus, there is another teaching demonstration. We're trying to hire 2 new anatomy and physiology faculty members. We had some a while ago. You can earn 3 percentage points on your first test. It's just a little, because I don't want you to be super motivated by just the points, but it ain't 0 on your first test by coming and filling out the evaluation form. We always ask students to come. The last stage of hiring faculty members here at SLIC is to ask for student and faculty feedback. We give them all the same topic. They teach head-to-head, and we let you guys help us decide, because you're going to be learning from them. And personally, I think that's very respectful of the learner. Nobody ever asked me about any teacher that ever got hired when I was going through college. So that's at 4:30 today. There's a form that you will fill out. It will go back to me. And if you already did it before, you don't get 3 more points.
How long does it take?
It's like 30 minutes. That's it. Go ahead.
Where?
TB 207, right next door. Right there. Yeah. So there'll also be another one next Thursday, I think, unless they drop out before then, at like 2-ish. That's when it's scheduled now. So if you guys want to come, I love to have your input. I always read every single thing that you guys write. Go ahead.
Can you go to the one next week instead of that?
Unless it's canceled, then yes. But the one today is not canceled. And if that person takes another job, then it doesn't exist. Okay? Okay. So, and then the other thing is I want to tell you guys about grading, because you're taking your test on Tuesday and you won't listen to anything I say while you're taking your test. So I got you captive right now. Okay? And that is that when do your grades show up? Where do your grades show up? When is you give me a week after you grade. It never takes me more than a week, but it almost always takes me a week, because your test has multiple choice part, a written part, right? And then it's also got a bonus part, right? And then I have to incorporate your quiz grades. So getting all of those together takes a little while. Those 4 scores make up your first test score. Canvas only lets me put 1 grade in per test score. So where do you see the breakdown? You see that on melanieb.com. And then once I've got you guys looking at the breakdown, you understand where it goes, then it goes up on Canvas. It's just 1 grade. And the other thing about Canvas is, if you notice, I put in a 0 for all of your final exam grades. All that is a placeholder, because you get to drop a test, right? But if I didn't put that as a placeholder, if you got a 70 and a 40, it would immediately drop the 40, and you'd think you had a 70 in the class, and you didn't have a 70 in the class if you got a 70 and a 40. Does that make sense? So I'm using that 0 as a placeholder for a dropped grade. So it doesn't mean that you've got a 0 for the final. It means it will give you a better indication of how you're doing on the class as we go through. Does that make sense? Canvas is kind of annoying. It doesn't really let you do very many things. I would like to talk to the programmer, please. I think that's it. And then I want to reiterate that the material that we're doing today is not going to be on your first test. It is directly related to this stuff, and it will reinforce some of the stuff that's on your first test, but it's going to be on your second test. Okay? So you're going to get the weekend to absorb all of the stuff that we've done. Okay, so first thing is we are going to, why does this set of notes follow the last one? Well, the logic to me goes like this. What we did is we set up cell transports up to the point that we could get understand how a cell membrane could be polarized, and it took a little while to get there. And then we took that polarized cell membrane so that we could actually learn how an action potential occurred, right? And we said that you can start with a graded potential, and then if you have voltage-gated channels, you can open the voltage-gated channels and cause an action potential. This set of notes is what happens when the action potential proceeds all the way to the end of the cell, right? Because we know that it does. You flip the first domino, it goes all the way down the axon to the axon terminals. And so what happens when you get the action potential to the axon terminals? Does anybody know? What happens? You release something. Yeah, you release neurotransmitters. So today is about teaching you the mechanism of releasing neurotransmitters and predicting what's going to happen when the neurotransmitter is released. Okay? So what we are going to be talking about is synapses and neurotransmitters in this set of notes. It's my favorite set of notes. It might not be your favorite. That's just because you're wrong. It's all right. Okay, so I like it. It's synapses and neurotransmitters are all about why you're smart or dumb or anxious or chill or artistic or ADD or ADHD or why you like drugs or why you like sex. It's all in the neurotransmitters. So I want to tell you something just so you don't freak out. FYI, look at the very end of this set of notes on the last page. This has neurotransmitters, receptors, chemical categories, functions, and drugs and disorders. Now, do you have to memorize all of those? If those aren't in the body of the notes, you don't have to memorize them, but people always ask me about the same ones. So this is a reference for you guys because you're going to be like, I want to know how cocaine works. Well, there it is, right? I want to know how this works. And this is because I am completely obsessive about researching neurotransmitters and drugs. As a matter of fact, I have literally, my husband's like, you're such a nerd. Because I have a spreadsheet that is a decade long and so enormous about all of the neurotransmitters we've discovered, all of the drugs and disorders. It's a thing of beauty. And my husband's like, what are you doing? I'm like, I'm working on my spreadsheet. He's like, oh my God, the hell? No, it makes me so happy. It's like organizing the universe. I'm like, it's all right. I got it. I understand. Everything's good. You know, like, so it's when all the stuff is in the right place. That's how I feel about it. Okay, so now let's start talking about synapses in general. So you've heard the term synapse before, and hopefully when you were taking anatomy, you at least learned generally what a synapse is, right? What is it, just in your words? Any idea? Yeah, it's kind of the site where two cells come together, but you have to be specific about what kind of cell. When you're dealing with the synapse, first off, one of the cells has to be a neuron, right? And so the neuron is going to almost, but not quite, touch the next cell, and the site of communication between the two of them is going to be a synapse. Now, synapses, there's different kinds of synapses. First, let's talk about what kinds of cells neurons can communicate with. Neurons can, of course, communicate with other neurons, which is what's happening in your brain right now, right? Communicating neuron to neuron. But are there any other kinds of cells that neurons can communicate with or command? What? I can't hear you what you're saying. Oh, muscle, yeah. Muscle and glands. All three kinds of muscle and glands, yes. Okay, so in this set of notes, what we're going to concentrate on is neuron-neuron communication, synapses. And the next set of notes, what we will be talking about is neuromuscular junctions, which is a type of synapse. So the synapses we're going to be talking about today are neurons communicating with other neurons. Now, you can communicate a bunch of different places with the neurons. Most of the time it's to the dendrites or the cell body. There's also this weird one right here that I'm not even going to talk about because it's super duper rare. It's hard to figure out how you could get an action potential if you had that one. So it's a big exception. We're going to kind of ignore it for right now. So we're going to do neuron to neuron communication, which is called a true synapse. In the next set of notes, what we're going to do is neuromuscular junctions, right? And the easy part, the good part about that is that you will already know how this part works. And all we have to say is the postsynaptic cell is going to behave differently because it's a different kind of cell, right? So synapses go before muscles, right? Okay, so we'll do neuromuscular junctions. There are also neuroglandular junctions in which a neuron tells a gland to secrete or something. We hardly ever really talk about those. They do exist. They're usually based on a similar electrical nature that these guys have. At least at the synapse. So synapses are the junctions between a neuron and a neuron or a neuron and an effector. And so what happens is the axon terminals almost touch the next cell. If you looked at it under a microscope slide, it would look like it's touching, but it's really microscopically separated. And that matters because what happens here is going to be really important. You could put the two things together with a gap junction, and then you'd have a really fast transmission between the two of them. But then you would be super limited in cell A and cell B would have to do the same thing. Does that make sense to you? So if you stuck them together with gap junctions, then they act as a unit, right? Do you want your entire brain to be on or off at the same time? Hell no, you don't. That is crazy. All of your brain cells on, all of the anxiety-producing cells on simultaneously, everything on at the same time. You don't want it to all be off either because that'll be the last day you ever had. So what we need is we need some cells on, some cells off. They can't all do the same thing. If they did, we would be using what are called electrical synapses, in which we stick together with gap junctions. You don't have those in an adult brain. We've never found them. You do have them in cardiac muscle, right? I can get an action potential to go from one cardiac muscle cell to another, or one smooth muscle cell to another. That's fine, right? As a matter of fact, you actually had them in your brain during embryonic and fetal development, which is kind of interesting. When you're putting together little Cletus the fetus's brain and you're getting all the parts in the right place, they just stick them together with gap junctions, right? And then as you need to start functioning differently, right? This cerebellum controls respiration, this does that. Then you pull them apart so you can control the cells independently. So it's like when you were putting the model together, you like stuck them all together with gap junctions as a placeholder, then you yanked them apart and started controlling them independently. So apparently embryonic and fetal brains do have electrical synapses. But what we are going to be talking about today is what's called a chemical synapse. And it's called a chemical synapse because I will use chemicals to communicate between this cell and that cell in the synapse. Yes? Not electrical synapse. So it's going to be called a chemical synapse. So what we are going to see is you already know some of this stuff, even though you might not have put terminology to it. Some of it you learned in anatomy, and some of it you'll just be able to figure out because you've been paying attention. First thing is, if I ever see two neurons in series, you can immediately tell which direction the information is flowing between them, correct? Which direction is it going? Yeah, it's going that way. Because you know and have known for a long time that dendrites receive information and axons send it, and then this one's going to receive it and that one's going to send it, right? So with that in mind, are you guys comfortable calling one of these the presynaptic cell and the other one the postsynaptic? Yeah? And of course, it's related to this synapse, because if I form another synapse down there, then you have to reclassify them, right? But we're going to say per synapse. This one is what? This one is presynaptic. Presynaptic cell, presynaptic neuron, presynaptic axon, it's all presynaptic. And this one is postsynaptic. Good. Okay, so I've got the presynaptic cell and the postsynaptic cell. And then what you are going to have is some more players that are going to get involved in there. So this, if it is between two neurons, and it is going to be considered a true synapse, neuron-neuron synapse. It could also be a neuromuscular junction if that one was a muscle cell, or a neuroglandular junction if that one was a glandular cell, but we're not going to do those right now. So let's talk about the functional anatomy of a chemical synapse. Okay, so first thing is, remember, dendrites and soma or cell body, axon hillock, axon terminals, yeah? And then the axon telodendria and the axon terminals. By the way, none of the textbooks can really quite agree what to call the ends here, but it's not particularly important. So here's my axon, yeah? Then it branches at the end. The fingers are called the axon telodendria, yeah, the fingers. Telo means end, dendro means fingers, so it's literally the fingers at the end of the axon, right? And then what about the bulby things? Because generally they don't look like this. They look like little ET fingers. Bulb, bulb, bulb. So, what are those called? Synaptic terminals, axon terminals, terminal boutons. Nobody can freaking decide. I don't care what you call them, because what is true is you need to understand their significance. I usually call them axon terminals. Okay, why are they bulby? Something is stored there. What is it? Yeah, this is where your neurotransmitters, and your neurotransmitters are stored there in neurotransmitters in what we call a synaptic vesicle. So this vesicle is like a membrane-bound bubble, right? And where was it made? Well, it depends on its chemistry. If it's a protein, it was probably made up here in the ribosomes. But if it was something else, you might be able to make it somewhere down there. Either way, they're stored there. You guys with me? Okay, so we've got our presynaptic cell and our postsynaptic cell right there. So now what I want to do is I want to set up a metaphor in which we start to understand how the presynaptic cell and the postsynaptic cell communicate. Just have a little bit more to define before we do this. First off, the term synapse is a bigger term than you imagine. This is not the synapse. The whole thing is the synapse, right? The site of the communication between the pre and the post is the synapse. There is a specific name for this space. What's the name for that space, guys? Between the pre and the post. What's the name for this space right here? I know you've heard it before. Yeah, it's a synaptic cleft, exactly. Okay, now, just some logic. Even if you didn't know before, what's the synaptic cleft filled with? Is it empty? Is it a vacuum? What's in there? Interstitial fluid, exactly. So can you diffuse through interstitial fluid? Yeah, of course you can diffuse through fluid, so keep that in mind. So synaptic cleft with interstitial fluid. Okay, good? All right. So what I'm going to have a situation is I'm going to set this up with a metaphor of pitcher, okay, catcher, ball, but the catcher, in order to catch the ball, needs a what? Needs a mitt. Now, the mitt in this metaphor is what? What will respond and bind to the chemical that is released? What are those going to be? Receptors. They are going to be receptors, yes? Okay. The postsynaptic cell can't do anything with a chemical unless it can receive and respond to the chemical, yeah? So we've got a pitcher, catcher, ball situation here. And then we're going to learn some more things about the receptor in just a second. So what we will have is a situation in which, let's go back to the last set of notes and let's put this together and then add just a little bit more detail. What is going to happen is, assuming that this cell gets enough chemical dumped onto enough chemical-gated sodium channels, are you guys with me? To cause a depolarizing graded potential big enough to hit threshold where? Axon hillock, right? Then I will get an action potential, and then the action potential proceeds very quickly down the axon. And then what happens is when it gets to the end of the axon, it will, by a mechanism I'm going to teach you in just a second, cause release of the neurotransmitter into the synaptic cleft, yes? And the neurotransmitter will go and bind to receptors on the postsynaptic cell membrane. Okay, now let's bring this story all the way back. So I got the pitcher, the catcher, the ball. Now, synaptic transmission across a synapse is one way. And one of the reasons you need to understand that it's one way is in this particular metaphor, the pitcher does not have a mitt. So even if the ball came back, you will not respond to it. You guys with me? So even if, for instance, this reuptook, I don't know how to say that word, took it back, right? It would be like rolling the ball back and you wouldn't get any response, not throwing it back. Does that make sense? Okay, so synaptic transmission is one way, in part, of course, because you don't have any receptors on the presynaptic side of the membrane, right? Does that make sense? Okay, so synaptic transmission is one way. In addition, I don't have an axon hillock here. I'd have a hard time getting it going, even if I could send it back. So there are multiple reasons, and I want you guys to think about this. So this note right here says that transmission across the chemical synapse is always one way, and it asks you why. So what I would like for you guys to be able to do after your test, because I know you're not going to think about this until after your test, is I would like for you guys to get 1 or 2 sentences that explains why synaptic transmission across a chemical synapse is one way, right? Okay, so we've got our pitcher, catcher, ball situation. And so now what we are going to do is we're going to add some detail, and we're going to figure out like how you actually release the ball, what the postsynaptic cell does when it gets the ball, and all that kind of stuff. So we're going to add some more detail to it. You guys all right with that? Okay, so interactive physiology is really good for this too, but you won't look at this stuff until later. So what it says is a neuron is separated from its neighbor by a space called the synaptic cleft. A similar space separates neurons from muscle cells. Here is something that doesn't happen. I don't know if you can conceptualize. It's always hard for me to ask this question in a way that comes up with the answer. One of these years I'll get it. Okay, what happens is, why can the action potential not jump across a fluid-filled space? So I can't actually have the action potential jump across the space. So if I wanted an action, I had an action potential here, and I wanted an action potential here, why can't it jump across the space? Anybody have any idea? What? Doesn't have a direction? I want you to think about what an action potential is. An action potential occurs on structure. What structure? It occurs on a cell membrane. So in the absence of a cell membrane, could you have a continuous action potential? No, you couldn't. Now, that's why if you stuck them together with gap junctions, you could have a continuous action potential, right? But if you don't have a cell membrane, what the hell would an action potential even be, right? There's nothing for it to have a difference across the cell membrane. There's no way to measure voltage difference between the two sides. So it can't jump across. So here's the deal. In order to get this one to have an action potential, what I have to do is I have to bridge the space with chemical messengers, and I have to start the damn story all over again with every single neuron. So you already told me this part. You told me that you need a bunch of chemical to open a bunch of chemical-gated sodium channels here, to hit threshold here, to get the action potential going that direction, right? Then I'm going to have to release neurotransmitters. How do I get an action potential here? The neurotransmitters are a chemical. Let's say that those neurotransmitters bound to receptors that were also chemical-gated sodium channels, yes? And that would what polarize this cell? Depolarize that cell. If you hit threshold here, that would cause another action potential. So what we do is we tell the whole damn story all over again every single time with every single neuron. It's not continuous, it restarts, which is why all that time that we put into understanding an action potential, now we get to do it over and over again until we're so damn bored with it. Now, what are we going to do today is we're going to figure out the mechanism of release, and we're going to figure out the different types of neurotransmitters and what kinds of commands you can give to the postsynaptic cell. Okay? So, the space is bridged with chemical messengers. What type of chemical messenger? You told me. Neurotransmitters. Now, let's back up to that first set of notes, and I want you to tell me what two criteria do you have to meet to be a neurotransmitter? There are two. What? Released by a neuron. That's one of them. Can't go into the blood. It has to be released close to its receptor or target cell, right? And this one qualifies, right? Okay. Released by a neuron close to the target cell. Okay. So what's going to happen is let's get the action potential down here to the end. What I want to talk about is how you release the neurotransmitters. What's the mechanism of releasing the neurotransmitters? So when the action potential reaches the axon terminal of the presynaptic neuron, you get to involve a new kind of channel that you haven't met before because it's only present at the axon terminals. We haven't really talked about those yet, right? So a new kind of channel that is only present at the axon terminal is called a voltage-gated calcium channel. And the voltage-gated calcium channel will open. What do you guys think? If you want it to open when an action potential gets to the end of the axon, should that voltage-gated calcium channel open in response to depolarization or hyperpolarization? Depolarization, right? And why will it open last? Well, it's because it's only at the end. That's why it's going to open last. So here's what happens. When you get to the end, you open a voltage-gated calcium channel, and then calcium goes down its electrochemical gradient. It's high on the outside, low on the inside. Of course, it's also negative on the inside, right? Negatively charged on the inside, so it moves inside which cell? Presynaptic or postsynaptic? Presynaptic cell, and that matters, right? It moves inside the presynaptic cell. And the increase in calcium concentration here is the trigger for exocytosis of the neurotransmitter, assuming you have ATP, because you can't do exocytosis without ATP, right? Okay, so calcium influx will cause exocytosis of the neurotransmitter. You guys remember how exocytosis goes. I had a bubble, membrane-bound bubble, a vesicle, push it up against the cell membrane, dump the neurotransmitter, keep the phospholipids, right? So that's what you end up doing. Does that make sense to you guys? So I dumped the neurotransmitter that was there into the synaptic cleft. Good? Okay. So now, what kinds of neurotransmitters exist? What kinds are going to be dumped? Until about 2 years ago, we only knew of neurons that made, stored, and released a single neurotransmitter. So if we said that this was a serotonin neuron, it makes serotonin, it stores serotonin, it releases serotonin. GABA neurons make, store, and release GABA. Dopamine ones make, store, and release dopamine. But you know what happens in biology when you make a rule? There's always a damn exception. Somebody found a couple of years ago a neuron that makes, stores, and releases 2 different neurotransmitters, and we have no idea how it decides which one to release. So we're just going to pretend that those don't exist for a little while because I don't know how the hell they do it, right? And nobody knows how they do it yet. So generally, you could understand this. If you got it stored and you're going to do exocytosis, you're like, oh, increase the calcium, release what I had here. So we're just going to assume that all of them are like that, even though put an asterisk in your head because eventually we'll figure out what the hell's going on with the other ones. So whatever neurotransmitter it had there, let's say it releases that one. And then the neurotransmitter is going to bind to protein receptors, and usually, not always, those protein receptors are chemical or ligand-gated ion channels. Yeah? Just like we were talking about before. Chemical-gated sodium channels or chemical-gated potassium channels. Chemical-gated sodium channels or chemical-gated potassium channels. Now, these receptors, in which the receptor is also an ion channel, the term for those is an ionotropic receptor. And they're really fast at changing the membrane potential of a cell compared to the next type that we're doing. Okay, you guys with me? Ionotropic receptor. Really, receptor ion channel is what it is. Receptor and an ion channel. Okay, so what's going to happen is the neurotransmitter is going to bind to and open a ligand or chemical gated channel, and it will cause a graded membrane potential in the postsynaptic cell now, right? So let's look here for a second. So what we did is we bound and opened that. So let's show that visual that we looked at last time, only this time we're going to pay attention to the beginning of it, okay? So last time we just kind of ignored the beginning of this one, but now we're going to pay attention to it. And we're going to say, okay, we've got an action potential moving down the axon. We're going to open a voltage-gated calcium channel. Calcium is going to move into the what synaptic cell? Presynaptic cell. The increase in calcium concentration will trigger exocytosis of whatever neurotransmitter you have there. That one's acetylcholine. Acetylcholine is going to diffuse across the what? Synaptic cleft and bind to ligand-gated sodium channels. Ionotropic receptors is what they are. And then sodium is going to move in this particular instance. And then, of course, that is what polarizing the postsynaptic cell? Depolarizing the postsynaptic cell. So that's what we've got going on right here. You guys with me? So we're just putting those two parts of the story together. Okay, so now, you don't always have to excite the postsynaptic cell. You don't always have to get it closer to an action potential. The reason you are not batshit crazy is because right now in your brain, and maybe you are, but I mean, you're sitting here, so you could be crazier, right? But the reason you are sitting here right now is because some of your neurons are on and getting action potentials, but some of them are off, right? And you've ever had that balance in which you're like, too many of them are on at the same time. Like, I need to give a presentation and I need for my prefrontal cortex to remember all of the smart things that I want to say, but my amygdala is telling me that I might die if I stand up. You really want part of your brain to shut up, don't you? You're like, shut the hell up. There's no time to think about that time that your pants fell down in 4th grade. This is literally no time for that to happen. But it's like, hey, remember that? You sometimes want to turn the postsynaptic cell on and sometimes you want to turn it off. So how do you do that? Well, what happens is it really depends on the neurotransmitter and the receptor that you are going to release and respond to. So let's say in this particular situation, I'm releasing a neurotransmitter that binds to and opens chemical-gated sodium channels. Am I getting closer to or further away from an action potential if I open chemical-gated sodium channels? Closer to, right? I might not hit threshold, but it's the right direction, okay? For that reason, we will call that synapse an excitatory synapse, yes? Okay, it's the right direction for an action potential, even if you don't get there. And so if the neurotransmitter binds to and opens a sodium channel, it causes depolarization of the postsynaptic cell and therefore excitation of the postsynaptic cell. This is called what's happening on the postsynaptic cell. This term shouldn't confuse you, but most people don't like it. What's happening on the postsynaptic cell if you bind to and open chemical-gated sodium channels is the postsynaptic cell is undergoing an EPSP. Excitatory, because that's what it's doing. Postsynaptic, because that's where it's occurring. And potential, because it's actually a graded potential, right? An EPSP, an excitatory postsynaptic, I say graded in my head, you don't have to, potential. Like trying to get you closer to an action potential, right? So the whole synapse would be considered excitatory. You might hit an action potential, you might not. If you didn't get one the first time, you could release more chemical and maybe get one the second time. Does that make sense? So you are exciting the postsynaptic cell. But how in your brain do you ever inhibit postsynaptic cells? When you want to turn off your amygdala from remembering the time your pants fell down in the 4th grade, how can you do that? Well, for most of the time, my amygdala just does whatever the hell it wants. It's like, oh, you just remember this. Remember, you're trying to sleep. And it's like, no, remember that dumb thing you said 2 years ago to that person who still hates you? You got to stay up and think about that. You can solve that now. Okay, but let's say we actually want to turn a cell off or down or make it less likely that it's going to have an action potential. What we would do is release a different neurotransmitter, right? So we would have, for instance, a different synapse. So stick with me. This one, let's say it only releases an excitatory one. But this one has more than one kind of receptor, right? So maybe I got a synapse over here that has the capacity to release inhibitory neurotransmitters. A neurotransmitter that binds to a different receptor, binds to, opens a chemical-gated potassium channel. Yeah? You bind to and open a chemical-gated potassium channel. Are you going to depolarize or hyperpolarize the cell? Hyperpolarize the cell. And is that exciting it, making it closer to an action potential, or inhibiting it? It's inhibiting it. That's one of the primary mechanisms of inhibition in the nervous system, is to hyperpolarize them so that they are not going to get an action potential, right? And if you hyperpolarize it really, really frequently, then you make damn sure it doesn't get an action potential, right? Shut up, shut up, shut up, shut up, shut up. I said shut up, right? Just shut up, sit down. Okay, that makes sense? Not you. You don't need to shut up. Okay, so that is an inhibitory synapse. So if the neurotransmitter binds to and opens a potassium channel, the result is hyperpolarization. You cannot use the same neurotransmitter on one cell to excite and inhibit it. It has to be 2 different neurotransmitters. So you would never know what the hell it was saying. So what we'd have to say is, let's say this postsynaptic cell was excited by acetylcholine but inhibited by GABA. So it would have an acetylcholine receptor that was a sodium channel and a GABA receptor that was a potassium channel. Does that make sense? So you can tell the cell a bunch of different crap, right? With a bunch of different chemical messengers. So if the neurotransmitter binds to and opens a potassium channel, the result is hyperpolarization and inhibition. And so we describe what happens on the postsynaptic cell as an IPSP, inhibitory postsynaptic potential. Right? Does that make sense? Okay. So some inhibitory synapses, by the way, don't use potassium. You can use chloride too if you want to, because chloride will go one direction and you can inhibit it. So let's just, for comfort's sake, assume that if I didn't tell you otherwise, you will use sodium channels to excite and potassium channels to inhibit. And then later on, you will figure out that there's some more, right? It doesn't matter how you depolarize or hyperpolarize. Depolarizing will excite and hyperpolarizing will inhibit. Just like regardless of what ion you use to do it. Okay, so let's take a little clinical connection break and make this relevant. Okay, so epilepsy. Epilepsy is a complicated disorder, and it could be genetic. It could be as a result of an injury. It could be a result of a past infection. It could actually be brought on by some medications. But what happens in epilepsy is that basically your central nervous system has an electrical storm. And what happens is you end up getting action potentials just roving through your brain like a hurricane, okay? And they could start in one location, and it ends up activating all of these other cells in other locations. And of course, if it happens in the brain controls the musculature, and you can get convulsions and seizures associated with it. Does that make sense? So epilepsy is a big deal. It's potentially life-threatening. You can get yourself in situations in which you can't safely take care of yourself. A lot of epileptics can't drive, for instance. One of the treatments that they can use for epilepsy correlates to this story. There's a lot of different possible treatments, but one of the early stage treatments they can use for epilepsy is that they can give you a drug that slightly hyperpolarizes all of your neurons. Can you see that that would make it less likely that you would have inappropriate action potentials? Yeah? It's called primidone, one of the drugs that does that. Compliance of patients on primidone is pretty low over long periods of time, because if it makes it less likely that you'll have appropriate action potential or inappropriate action potentials, it also makes it less likely that you'll have appropriate action potentials. So what happens is you end up feeling both mentally and physically sluggish, right? You're like, I I've got brain fog. I'm exhausted all the time. I can't think of things that I used to be able to think of. My reflexes are really slow. You get really exhausted, and you're tired of being dumb as a stick and exhausted. And so what happens is they have a bunch of seizures. They say, okay, I don't want to have these seizures. These are really dangerous. But they go on primidone. Seizures go away, but they feel dumb as a stick, right? And a lot of times depression comes along with it because you're tired of being stupid and tired. And then they go off the medication, and then seizures come back, and then they go back on the medication, and then seizures go away. And so that cycle a lot of times happens with people who are epileptic, but it's all based on hyperpolarizing the neurons. Now later on, there's some other interventions. There's different drugs and other interventions. They can use electrical stimulation now on the brain that actually seems to help with epilepsy. They don't even know what it does, but it works. A lot of the things that they're doing with your central nervous system, it's like, ooh, take this. Oh, that worked. One of the drugs that's like been used for decades with the central nervous system is lithium, and they have no idea how it works in there. And then later on, if the epilepsy is so severe that it's not controllable by almost any other mechanism, sometimes what they'll choose to do is slice your corpus callosum so that an epileptic that started in a single hemisphere wouldn't be able to travel over to the other hemisphere. Does that make sense? Okay, so now, there is another type of receptor. I'm not going to show you a picture about it right now. I'm just going to tell you that it exists, and we will see them when we get back to endocrine system. So the fastest way to depolarize or hyperpolarize the postsynaptic cell is using an ionotropic receptor. Because the chemical binds to it, it opens it, and changes the membrane potential. There is a slower way of doing it that's actually not uncommon in the brain. And it's called a metabotropic receptor. And instead of an ionotropic receptor in which you take the neurotransmitter, you bind to the receptor, and it opens the ion channel. Imagine that one is like playing pool. And so what you're going to do is take the neurotransmitter, and you're going to bind to and then shoot it straight into the pocket, right? And you're going to start that action relatively directly. So the cue ball would be your neurotransmitter, and the ball that you hit straight into the pocket would be your actual ion channel, right? You made it happen very effectively, as quickly as possible. But you could choose to be super duper tricky and do a 4-ball combo shot, which is going to be slower, right? And what's going to happen with that one is you take a neurotransmitter and you bind it to a receptor. The receptor is not the ion channel. The receptor bumps into 3 more proteins, and the 4th one is the ion channel, right? That's called a metabotropic receptor, and those are slower. They are not for immediate responses in the brain, but they sure as hell do exist, which is so weird to me because I'm like, how the hell did you evolve something that has 4 proteins involved, right, when you could have 1? And how, I don't even know how the hell that survived in the evolutionary pressure. It's amazing to figure out, but they do exist, both for hormones and for neurotransmitters. Okay, so regardless of the type of receptor, let's say you set off a new graded potential on the postsynaptic cell. Let's say it's a depolarizing graded potential. If it's big enough, it's going to cause that one to hit threshold, and then you're going to have an action potential on that one. But here's a concept that the last class had a little trouble understanding. Hopefully you guys will get it. Okay, so let me see if I can present it this way. This cell has to be responsive to frequency coding, right? It means that it has to tell the difference between 1 neurotransmitter release and 25 different neurotransmitter releases. Does that make sense to you? So if I release the neurotransmitter and let it stay in the synapse, then I wouldn't be responsive to the difference between 1 and 25. Does that make sense to you? So what you have to do with neurotransmitters is you have to release them, and then you have to immediately remove them from the synapse so that you can understand frequency coding. If you left it around, you couldn't tell the difference between 1 neurotransmitter release and 25 neurotransmitter releases, right? So what we have to do with neurons is we have to cause a response and then remove the neurotransmitter from the synapse so that we can tell if there's another neurotransmitter release. So you have to release them, but then you have to get rid of them from the synapse. There are 4 ways that I know of getting the neurotransmitter out of the synapse. The least efficient one you're going to see right here. The least efficient one is to just let it diffuse away. You better not be in a hurry, right? That one's not going to be super responsive to frequency coding. And some places in your brain don't need to be super responsive to frequency coding. So you could let it diffuse away. The second way you could do it is, look here. you can reuptake it back into the presynaptic cell so that you can reuse it. You'll reuptake it, you'll repackage it, you'll reuse it. Now, that is not going to cause a presynaptic cell response because this is generally a protein, a transport protein. It's not a receptor. So with my mind, that's like the catcher rolling the ball back, not throwing it back, right? It's rolling the ball back to the pitcher so it can be reused. Does that make sense? So you can reuptake the neurotransmitter so that you can re-release it, okay? The third way that you can do it is that in some synapses, by the way, this is representing all of these mechanisms in one synapse. They don't have all of them. It's got one of them in a synapse, okay? The third way you can do it is that the postsynaptic cell has an enzyme embedded right there that will break down the neurotransmitter after it's used, so that it won't cause a postsynaptic cell response more than once. So what would happen is the neurotransmitter would bind right here, that's the receptor, and after it released, it gets broken down by this enzyme. For instance, acetylcholine is most commonly, after it's used, it's broken down by an enzyme called acetylcholine esterase into acetic acid and choline. And separately, they don't cause a postsynaptic cell response. And so what happens is the presynaptic cell then uptakes the most precious part, which is choline. It's hard to get enough choline, right? So it reuptakes the most precious part. So it breaks it down. As soon as you change the shape, it won't activate the postsynaptic cell receptor anymore, right? Okay, everybody good with that? And then there's one other way that isn't pictured here, and they only figured it out a couple of years ago, and I think it's kind of cool. And that is that they have figured out that the neuroglial cells called astrocytes, remember learning about astrocytes? Astrocytes can act like ball boys. So what they can do is, if I've got two neurons that are communicating right here, the neurotransmitter was released and it caused a response. Remember, astrocytes are kind of around neurons, like scaffolding and holding things in place. What the astrocyte will do is reach in and grab the neuron and then hand it back to the pitcher, to the presynaptic cell. And we didn't know that before. We only figured it out because some people started getting astrocyte disorders that caused problems with neurotransmission, and they couldn't get adequate levels down or up in the synapses. Does that make sense to you guys? So those are the 4 ways of doing it. We will run into all 4 ways as we are moving through. So the neurotransmitter can diffuse away. An enzyme can degrade the neurotransmitter. And 2 enzymes that you'll run into are acetylcholine esterase is usually used to break down acetylcholine in synapses. And then monoamine oxidase is an enzyme that breaks down norepinephrine, epinephrine, and dopamine. And we will see that the reason you have a similar breakdown and also reuptake mechanism for those 3 is because they're almost exactly alike chemically. They're like chemicals that are super close to one another. So you can use like the same reuptake mechanism or the same breakdown mechanism. And then sometimes the astrocytes will help with this. And then sometimes it's reuptake. So let's talk about the reuptake because this one has actually touched your life. I know it has. So reuptake, in which the whole neurotransmitter is reuptaken into the presynaptic cell so that you can release it again. So let's talk, and this won't be the last time that we talk about it. Let's talk about antidepressant drugs and a class of antidepressant drugs that actually use this reuptake thing. So antidepressant prescriptions are incredibly high in Utah, incredibly high compared to all of the other states and even controlling for population size. People argue left, right, and center as to why that is, and I got my opinion and you got yours. Mine is because you need a damn cocktail, honestly. You have to medicate yourself every day if you got a cocktail every day at 5:30. Some people say it's the altitude. We've been the same altitude for a really long time, dudes. Some people say it's the pollution. Some people say it's whatever. You can argue with your family about it at Thanksgiving. But regardless, we have really, really high rates of antidepressant prescriptions here. So let's talk about how the most common category of antidepressants work and what they have to do with this. One of the things that can manifest in depression and anxiety is low levels of serotonin in some areas of the brain. Okay, interestingly, it tends to manifest as anxiety even more frequently in women and depression even more frequently in men. And we don't know why that is, but regardless, depression and anxiety has to do sometimes with low levels of serotonin in certain regions of your brain. Now, nobody ever goes at the doctor when you're depressed and tests your serotonin level because they are released in synapses and immediately reuptaken. They don't circulate in the bloodstream. How the hell do they know anything about your serotonin level? So they're really just experimenting on you, right? And you should be involved in the experimentation process. So I want you guys to know what they're trying to accomplish here. So if low levels of serotonin can be associated with depression and anxiety, if we can trick your brain into having more serotonin or thinking it has more serotonin, maybe you will get relief from the symptoms of depression and anxiety. So I want you to imagine this picture that we've been looking at is a serotonin synapse, okay? And so what I want to do is I want to increase my level of serotonin. Now there are ways to increase your level of serotonin, but most of them involve work that people are not willing to do. If you're living with a jerkbag, probably move out. If you freaking hate your job, see if you can quit. But that's privilege, right? Sometimes you can't quit your damn job. If you are super depressed and anxious about things that are happening, you can change them. You can also have more sex and exercise more, and that will increase your serotonin level. But most people are like, that sounds like work. I would like a pill, because I ain't got time for all that. So now they're going to give you a pill, and the most common category of antidepressants are SSRIs, selective serotonin reuptake inhibitors. They better be selective because serotonin, in addition to controlling mood, also controls appetite, controls weight gain, controls sleep, makes you hallucinate. All of the hallucinogenic drugs are serotonin-based. So we need it to be selective for the synapses in my brain that are related to depression and anxiety. So here's what it does. Take an SSRI, Paxil, Prozac, Zoloft, a bunch of them. Those will inhibit the reuptake of serotonin from selected synapses, right? So what they are going to do is if serotonin gets to stay in the synapse longer, then your brain interprets it as being releasing more serotonin. Yeah? So it's kind of like you're tricking it into thinking you're doing frequency coding, right? Like, I'm happy. No, I'm happy. I said I'm happy. When basically you release it once and then it just gets to stay in the synapse longer, because what we have done is we have figured out specific reuptake proteins and how to block them, right? So it stays in the synapse longer and it gives you potential relief of depression and anxiety. Does that make sense? That's how this process is related to this stuff. So now, not all antidepressants, just because we've got some time and you guys are probably interested in this. Not all antidepressants are based on serotonin, because not all of your happiness, sadness, depression, anxiety is based on serotonin. So these are your three happy chemicals. They are your three favorite chemicals, but you just don't know it, right? So typically what serotonin is responsible for is kind of mood regulation. Serotonin is the reason that, like, when you get a flat, you don't break out into tears, right? And when something happy happens to you, you don't go completely manic and not be able to get down. It's like, hey, I'm happy, but I'm not losing my crap over it, right? But dopamine is my favorite one. Dopamine is your reward neurotransmitter. Dopamine is like, that was amazing, do that again, over and over again. And so that one, if you have dopamine, you tend to have pleasure, right? And then the other one is your up chemical. It's like, I can do this. And that is norepinephrine, alertness, concentration, and energy. So what we would really like is to be in the middle of that Venn diagram in which we've got perfect levels of all three. We're happy, we're up, we can regulate our mood. I think I've been there twice in my entire life, right? That magic little portion of the Venn diagram in which you've like got a good mood and also you're firing on all cylinders, you're getting all your crap done and nobody's pissed off at you. But that is not how life quite often goes. If you have two and not the other, then they tend to manifest in these strange ways. The other really common antidepressant drugs work on the other two. So SSRIs are usually the first tier antidepressant drug, and I've taken all of them because I'm crazy and I'm just constantly trying to figure out, like, I didn't get a user's manual with this thing, so we're just going to see what happens. So the SSRIs work on serotonin. There's another category, the SNRIs and SDRIs. For instance, Wellbutrin is one of those. Wellbutrin works at dopamine and norepinephrine synapses, and it actually inhibits the reuptake of dopamine and norepinephrine in those synapses. So it's a different type of antidepressant. Does that make sense to you? And it feels different too. It's more up, it's more happy, not necessarily super regulated, but more up and more happy. Which one do you think is more likely to cause you sleep problems, an SSRI or the Wellbutrin? Wellbutrin can cause sleep disturbances. Which one should you definitely not take if you're not depressed, but you're super anxious with a predisposition to panic attacks? You shouldn't take the norepinephrine dopamine one, because you will be super happy except for the moments in which you're crawling under the table and having a panic attack. So it's contraindicated for anxiety disorders with panic attacks. So who knows which one you need? And the interesting thing about this is literally no one. No one ever tests your levels of neurotransmitters. You go in and you self-report your symptoms, right? And they say, try this, and you try it. And then if you are a really, really well-educated patient, what I do every single time I try a new drug, and I never let them tell me which drug, I tell them which drug I want. Because I've like done all the physiology research on it. I'm like, oh, okay, I want this one. I think this one might work. Then I take notes every single day about how I feel and how much I slept, right? Because I love Wellbutrin. I'm super perky on Wellbutrin, but I can't sleep for shit. I just cannot sleep at all. And then eventually I just start losing all my crap. You know what happens when you don't sleep. You're like, oh, everything's falling apart. I had 2 amazing weeks and now everything's falling apart. So basically I'm on nothing now. This is just me. It's just like how it is. Because there is no perfect drug. There just isn't. I want you guys to invent one, okay? Because you'll be super wealthy and I'll be happy about it. Okay.
Will you try them for us?
Hell yeah, I will. I'm an experimental human, man. I only get one life. I might as well die with some good stories. Okay. So the last thing that I didn't tell you guys, and I'm going to let you guys go a little early today because the last class was a little behind you, is that, so we've been talking about synapses. What I want to tell you is some cool stuff that they've learned about synapses. So one of the cool things that we've learned about synapses is that there is a direct relationship between what you do with your brain and how many synapses you actually form. So I know everybody's told you over and over again that for all practical purposes, your neurons are amitotic, like you don't really get any appreciable number of new neurons after fetal development, right? That's why you get one shot at like making a fetal brain, and then it's just how it is. But what isn't true is that you can't, it is true that you can make new synapses. And so what we have figured out is there is a relationship between synaptic formation and memory formation, which I think is kind of funny because we have been saying for years that when you understand something, you make a connection, right? And it turns out that that's literally what's going on in your brain. What is going on in your brain is when you're like, oh, okay, osmosis. You taught me osmosis 3,000 times. And then you go, oh, okay, this is why I shouldn't drink salt water. And you get that aha moment, and you're like, oh, okay, and you make the connection. What the hell is happening in your brain is that one of these neurons that you're seeing right here is branching off a new branch off of its axon, yeah? And it's actually going and connecting to another neuron. So you are changing the physical structure of your brain by making it learn and experience new things. So it actually changes the white matter connections in your brain, what you actually do with it. So we've got this evidence of synaptic formation. It's called synaptic plasticity. And the more you do with your brain, the more synaptic formations you get. They start to prune as you age, but the more you have, the more you have that you can afford pruning on. And then they even have some cool evidence that this is actually the case in some gross anatomy studies in which you actually section a brain post-mortem. So one of the things that we have figured out is that post-mortem, you can look at the white matter. The corpus callosum is white matter. It would be myelinate axons, right? Which would be an indication that synapses had formed on either end, right? Okay. So if we look at the white matter of some brains versus other brains, what we can see is that people who had certain types of activities when they were alive, especially if it was extreme, how much activities they did, have a thicker corpus callosum, immeasurably thicker, not even microscopically thicker, like measurably thicker corpus callosum. Do you have any idea what activity dramatically changes the thickness of your corpus callosum? Any idea? No, it's actually music.
Really?
Music training. Music is really interesting because what it requires you to do is it requires you to do the pattern formation parts of your brain. You also have to write music, right? You use the creative portions of your brain. You use the emotional centers of your brain. You attach it to the motor control centers of your brain. And all of it requires so much back and forth between the two sides of your cerebral cortex that post-mortem, if you look at the corpus callosum of a lifelong musician who is not just a player but also a writer, their corpus callosum is measurably thicker than the rest of us, which is why my poor little 11-year-old has been in freaking music lessons since he was 4. Like, I hate piano. I'm like, why did you change the world? You need a thick corpus callosum. He's like, I don't know what you're talking about. Okay, we will stop there for today. I let him switch to drums this year, though, and drums is way cooler. Apparently. He says he's never going to play piano ever again. I'm like, some girl's going to think it's amazing, and then you'll play.